Hello viewers, today I will discuss about the effect of mutation at chromosomal level. I will talk about their structural and numerical change regarding types, phenotype and mechanism. As we know that change in DNA sequence coding for gene in chromosome that can be happen in any part of the chromosome causes physical rearrangement that gives the structural abnormality as well as that change the numerical balance. First, in 1916, by Brebner-Robertson, the Robertsonian translocation in grasshopper was found. Barbara McClintock have found during 1940s the cytological evidence of chromosomal abnormalities in maize. Bridges have found the X chromosome related deficiency in Bar locus, Bar I phenotype. So, as characteristics, it is a gross or large-scale mutation which can be cytologically observable, that is related to the quality as well as quantity, and translocation, duplication, deletion, inversion all are included here. Significance, sometimes it is related to the birth defects as in found in case of Down syndrome for extra copy of chromosomal 21. But in case of plant, it is related to the evolution because the new plant species are evolved by polyploidy. Types, the chromosomal qualitative change that is found in case of structural change as well as chromosomal quantitative change related to the numerical change. The first one, chromosomal structural change. Now, structural changes are related to the breakage. Then after breakage, the fates may be number 1 ununited or restitute. That broken part may be added to any homologous or non-homologous chromosome. And number 3, non-restitutional union. So, breakages may come from transposable element, chemical mutagen or irradiation as well. Now, types, usually these may be the qualitative as well as quantitative. The qualitative includes the inversion which may be paracentric or pericentric. Quantitative includes the duplication, translocation as well as deletion. Duplication may be tandem, reverse tandem or displaced. Displaced may be homobranchial or heterobranchial. Translocation may be reciprocal, non-reciprocal or shift or intercalary. Now, these non-reciprocal may be simple or Robertsonian. Now, deletion, again it may be terminal or intercalary. The duplication or insertion characteristics. As we know that any section or part of chromosome can be added to the other portion, other location of the chromosome. So, any segment is getting duplicated or inserted and we know that the fragment is getting duplicated here. The types include the tandem, reverse tandem as well as displaced. Tandem means in the same direction, reverse tandem means in the opposite direction. Displaced may be on the same arm that is called as homobranchial or on different arm that is called the heterobranchial. Next, mechanism. Usually, it is done with unequal crossing over during synapses. So, that may not be joined to just the neighboring portion, any different loci, any different chromosome, the duplication can be done and usually duplication is done by bridge breakage fusion cycle. So, dicentric chromosome on 2 pole make the bridges and bridges can be broken at any point and ends are getting fused and this cycle can go on. Phenotypic effect, that may be dominant or recessive or sometimes may be intermediate inheritance can be happen, but in all the cases they are the cumulative effect. That was found with respect to the Bar B locus on the X chromosome of Drosophila where with extra duplication the size of the Bar I is getting reduced. Not only that, the gene effect mutation, the effect of gene duplication is found related to the aldosterone synthetase of steroid synthesizing genes. We can find the duplication of these genes gives the high blood pressure. So, along with old capabilities, new capabilities come. Not only that, in case of Huntington disease, the CAG repeats are found, trinucleotide repeats, and in case of CGG repeats found by the disruption of the FMR1 protein in case of Fragile X syndrome. But when we are talking about the duplication of the chromosome, just one set can cause disorder like Down syndrome, chromosome 21. But if the sets are getting multiplied polyploidy found in plant causes the beneficial effect. Next one, the characteristics of the deficiency or deletion. So, some part of the chromosome can be detached or deleted containing the genetic material, that may be DNA sequence, single base or block of chromatin from the chromosome. Types may be the terminal deletion, that is the end can be deleted, but the broken end can be healed very easily. Or maybe the intercalary, that is the inner part of the chromosome can be deleted by 2 breaks and then after break they will be reunited. Mechanism, usually mechanism is done, suppose some centromere portion is getting lost and that particular lost portion without centromere will fail to make the spindle during cell division. So, that will be lost by cell division. The causes may include the 3 factors, maybe the loss during translocation, the unequal crossing over as well as the breaking without the rejoining. That everything, this terminal deletion, avoid to them, some looping may happen sometimes and this chromosome section may lose some gene until and unless they are united with any other chromosomal portion. Next one, phenotypic effect. Usually these missing effect of the chromosome give the bulking effect as well, and some portion of the genetic material is lost, and on what portion is getting lost, that actually give the particular phenotype. We can have different types of the phenotype in case of Drosophila, like pale, beaded, notch, etc. And notch has pseudodominance over the facet loci or phenotype. The sex ratio is getting changed and suppression of the crossover is also found. In case of human, we can find the Cry-Du-Chat syndrome by the deletion of the chromosome 5, and we can find Philadelphia chromosome is lost, chromosome 21, and in maize the chromosome 9 lost gives rise to differential phenotype. In case of prokaryote as well, like E. coli and lambda fudge, this deletion is observed by the collapse of single-stranded chromosome by giving bushy structure, bush-like loop structure as well. Now, chromosomal translocation. The chromosomal translocation means change position of the chromosomal section to another location or interchange in between homologous or non-homologous chromosome. So, this can be done spontaneously or with the help of ionizing radiation. Types. First, the intrachromosomal and interchromosomal type. In intrachromosomal type, that is non-reciprocal. That means some portion can be detached and added to the another location, that cannot be exchanged. But in case of the interchromosomal translocation, the exchange happens in between 2 homologous or non-homologous chromosome. Next, the various types, that includes the reciprocal and non-reciprocal translocation and as well as shift. In case of simple non-reciprocal translocation, we can find one single break occur, some portion may be lost and some portion can be united at another location. But no losses are found in case of reciprocal translocation because 2 breaks are made in between 2 homologous or non-homologous chromosome and 2 portions are getting interchanged or recombined. And in case of shifts or intercalary translocation, 3 breaks are made. In one chromosome, 2 breaks are made, and the part is getting deleted and added to the single break made on other chromosome. Non-reciprocal translocation contain Robertsonian translocation where whole arm is getting fused by centric fusion to other chromosome. Let's talk about that non-reciprocal Robertsonian translocation. That may be found in between 2 non-homologous or homologous chromosome. That was found by the Brebnan-Robertson in 1916. And it was found that in these cases, 2 acrocentric chromosomes are there, breaks are made, long arm fuse with another long arm. Short arm may fuse with the short arm, but without gene they can be deleted out. Mechanism. In all those cases, we have found that it is in case of heteromorphic sex that can make interchange in between non-homologous chromosome as well. And we can find in translocation, usually in case of these Robertsonian translocation, the balanced gametes are maintained, no lethal effects are found. But unbalanced combination of the genetic ratios are formed, and first after making the interchange in between 2 chromosomes, the first can do the with the third one, that is with the third chromosome, second translocation may form by formation of ring or chain, and it may be related to the deletion and deficiency as well. Genetic consequence, we have already seen that it gives rise to different segregation type like adjacent 1, adjacent 2 as well as alternate. Not only that, the linkage between the genetic loci is also changed, but balanced ratio are found in case of the Robertsonian translocation. We can find by translocation the disruption of the gene, or we can find sometimes gene may come under different promoter or enhancer guidance in case of Burkitt lymphoma, as well as we can find the production of hybrid gene, C-terminal of one protein and N-terminal of other protein by fusion, like that was found as phenotypic effect in chronic myelogenous leukemia CML by the production of hybrid gene BCR-ABL. Apart from these, we can find the multivalents are produced, that was found in Drosophila onthera, etc., as well as multiple translocation system was found in Onthera lamarckiana. We can find spontaneous translocation as well as crossing over is suppressed because lost during alternate segregation. Next one, chromosomal inversion. Some portion of the chromosome is getting detached, and by 180-degree inversion in the reverse way, it is getting reverse orientation, it is again reinserted, and these can increase the threat and causes the mutation and breakage as well. Now, types that may be paracentric, not including the centromere, or pericentric, including the centromere. In both the cases, they actually suppress the crossover and new gene orientation giving rise. Mechanism, as we know that the anaphase 1, it makes some sort of anaphase bridge-like structure, and bridge are broken, and from acrocentric chromosome to metacentric chromosome may give rise. So, in this way, the inversion may happen, and it causes some sort of restitution, some change. We can find the overlapping inversion in case of arrowhead and peak combined phenotype, as well as the breakpoint in the inversion can be detected in C3b stalks of the Drosophila. Next one, phenotypic effect. As well as we can see the gene order of the linkage is getting changed, sterility is found, and the suppression of the crossover. It is related to the balanced lethal system. So, what is balanced lethal? Suppose if two recessive lethal phenotype exist on two homologous chromosomes at different loci. So, the enforced heterozygous condition is made by the suppression of the crossing over with this inversion, because at the homozygous condition the lethal will be died. Chromosome structure in different systems. It was found in case of maize by Barbara McClintock, all the things, that is deficiency, duplication, translocation, deletion, everything. But the thing is that the chromosome polymorphism was found in case of various other aspects. First, the polytene chromosome in Diptera. It was found by 1881 by Balbiani, found the coil bodies, and the cytological maps were done with respect to these organization or reorganization of the chromosome, like deficiency, duplication in case of Drosophila X chromosome, and the cytological map of the genetic linkage was done. That actually include the chromosome polymorphism of B chromosome. B chromosome is also called supernumerary chromosome. Actually, it inhibit the homologous pairing in case of allopolyploidy. In case of microchromosome, we can find very short chromosome. Okay, that is actually found in case of reptile, fish, etc. And mini chromosome having the telomere, centromere, as well as replicating region, very small thing, can also be found in case of maize. The B chromosome is found in plant and fungi, apart from which the lamb-brushed chromosome is also found in amphibia, bird, etc. The coiled cable-like bodies, which actually help to know the chromosomal organization as well. The change in chromosome number. The change in chromosome number again by break and reunion may happen by centric fission or fusion. From fission, from two homologous chromosomes, they will be added at the centromere, or by fission, they will be split out into two chromosomes. That may be done by the recombination with respect to the tandem repeats and breakage and reunion. Types The two types are there, euploidy and aneuploidy. Euploidy may be monoploidy or polyploidy. That polyploidy may be allo or autopolyploidy. Now, aneuploidy may be hypo or hyper. Hypo include the monosomic and nalosomic, and hyper include the disomy, trisomy, double disomy, double trisomy, etc. Euploidy. In case of euploidy, just the sets of the chromosome are getting added or deleted, maybe from the same set or other sets. The type monoploidy. Actually, monoploidy may happen in gamete cell, not in somatic cell, and no segregation happens. That may also called as haploidy. That may generate spontaneously. In case of microorganism, that can be taken as the test for the genetic tool. X is equal to N. That is the single one, single set. But polyploidy is getting started from the diploid, extra. That is 3N. Triploidy is 3N. Tetraploidy is 4N. Pentaploidy 5N. Hexaploidy 6N. Octaploidy 8N. Now, generation of the monoploidy, usually monoploidy are generated by the irradiation of the pollen, and the reduction in the gametes of the diploidy or polyhaploidy can also be found. It is actually spontaneous haploid give rise to gene mutation, and irradiated pollens are used during the course of plant breeding. Types of polyploidy. Polyploidy may be autopolyploidy. The sets are coming from the somatic doubling of the same species. That is found triploid banana actually was found, seedless banana. And what we can find, the allopolyploidy, the sets are coming from different species and generated by unreduced gametes. The primrose, fertile primrose, hybridization of primrose is the example of allopolyploidy. And mixed polyploidy is found as autoallopolyploidy. The cytology of the polyploidy, actually it gives the multivalent structure. The multivalent structure can be resolved in any types of trisomy, triploidy, tetraploidy, etc. And we can see it can be observed in the pachytene stage of the pachytene chromosome, the pachytene association in the diakinesis stage. And it was found in brassica, tomato, tobacco, etc. Mechanism that can be done by the fertilization of one egg with different sperms, the fail in mitosis and meiosis, as well as irregular unequal segregation was also found, giving rise to isochromosome V-shaped structure. Polyploidy animal was found related to the sterility of the salamander, as well as in brine shrimp it is found related to the parthenogenesis. But giant cells, this sort of nucleus containing, was found the human kidney and liver as well. Polyploidy in plant, very good reasons are there. Triploid plants are found, and these triploid plants are generated from the tetraploid 4N and gametes created by the 2N diploids, and that is the example are forest trees, seedless watermelons, etc. Tetraploid wheats are found by generation of 2N diploids together. Hexaploid and octaploid wheat are found, and amphidiploid nicotina new species can be generated. Now, application of the polyploid, as we know that induced polyploidy can be created. An application, it is in actually happen in case of plant breeding. Different type of the triticum aestivum, triticum monococcum, dicoccum, having the 28, 42 chromosome number where X is equal to 7 can also be obtained, 28, 42 as well as 14. Not only that, the New World cotton is very high vigor, and we can find the primrose hybridization which is fertile, as well as we can find the tobacco mosaic virus, disease resistance tobacco. We can find polyploid fruits like Macintosh apple. Now, genetic consequence, actually the hybrid thing give the better phenotype, and genetic linkage over the chromosome can be mapped very easily. Genetic diversity can be done. The more complex structure can also be found, and rapid chromosomal segregation happens here. Next, aneuploidy. Aneuploidy means just single chromosome is added or subtracted, maybe from the same or different set. Trisomy, addition, gain. Monosomy, that is loss from there. Next, aneuploidy types. Now, these aneuploidy types may include the different hyper or hypo. Hyper means increase from the 2N diploid. That may be, or increase along with 2N, right? That may be trisomy or tetrasomy. Trisomy means 2x plus 1. We can find tetrasomics 2x plus 2. But hypo means the reduction of 1 or 2 chromosome from the 2n chromosome. That may be nalosomic or monosomic. Now we can find the triplotrisomic as well, 3x plus 1, tetramonosomic 4x minus 2, tetrasomic 4x plus 2, etc. All these combinations are found. Mechanism, they are coming from spontaneous non-disjunction, or they can come from the irradiation as well as variation in translocation deficiency causing the reduced fertility or viability. Synthesis, the aneuploidy can be synthesized by effect of ethyl methane sulfonate, by giving colchicine in sorghum in this way, or can be reduced from the unreduced gamete as well. Aneuploidy in human, trisomy is found in case of Down syndrome, that is 47 plus 21 chromosome found by Longdon Down in 1866. The abnormalities, congenital abnormalities are there. Trisomy 13 is called Patau syndrome, 47 plus 13. Trisomy 18, 47 plus 18, Edwards syndrome. The first one is related to the deafness and second one is mental deficiency, Klinefelter syndrome. That is the trisomic with respect to X, 47 XXY, that was established by Klinefelter in 1942. 47XYY behavior is also found by Jacob in 1965, and Turner syndrome, that is again monosomic with respect to X in female, 45 X that is found, along with 44 chromosome, 1 X is only present. It was found in 1938 by Turner. The females are there with abnormalities. In Drosophila, Bridges found the aneuploidy by non-disjunction, XXX metafemale and XXY is found. Aneuploidy in plant, Datura species, 25 chromosome, that is along with 24, 1 chromosome is present. The chromosome linkage is mapped by the nalisomic stalks, okay. So it helps the dominant mutant gene in tobacco to find out. Genic balance, always in Datura species or in other species, there are certain amount of gene at a particular location to cause a phenotype. Is the amount of gene is getting changed by dosage effect, the phenotype can be shifted, either maybe give the intersex or other. Chromosomal mosaics. So in case of chromosomal mosaics, what is getting happen from one particular state, from one particular organism, we can find two different cell line having different karyotype, okay. So one may contain the reduced chromosome, other extra chromosome. So various types of mosaics and chimeras is found. In case of mosaic, some portion of the area in case of body is actually found. What is going on here? Different mosaics, X by XX, X by XY, and various other combinations are found. Bar body, by Leon and Russell by Leon hypothesis, we can see one X chromosome within XX is getting inactivated. That is called the heterochromatinization. It becomes inactivated, okay. And this inactivation can be monitored by glucose 6-phosphatase dehydrogenase activity, the enzyme related gene over the X chromosome, and the dosage compensation, dosage effect was found in Drosophila. Position effect. So position effect means suppose the gene related to the genetic order, the phenotypic change can occurs related to the phenotypic change and genotypic change was found by the Strudvent. The nomenclature, usually the chromosome positive bending is found in Turner syndrome. The chromosome positive is found in Klinefelter syndrome where trisomic of X. In case of Turner syndrome, the chromosome negative is found. Nomenclature is that suppose 46XY1Q, it designates that one chromosome Q arm have the addition, plus or minus give the addition or deletion, and in case of 47XY14P plus shows the 14 chromosome is added and addition also happens in the P arm. So students, I have discussed about different types of structural and numerical change of chromosome and their different aspects. Thank you.